So I would like to discuss today the redundancy principle and application in cell biology. So let me first define what I meant by this principle that we introduced a couple of years ago. So let me start with a definition. By definition, the redundancy principle states that it is the possibility of having many identical copies of the same object at the same time. And the reason for this is to accomplish a task that can be performed by any of these elements. So basically, imagine you have the same copy, here represented by this point, of the same object that appear more or less at the same time. And the question is, why do you need so many of them? So suppose you have a certain number of them, like molecules. This can be molecules. It can be cells. It can be ions. And so there is a reason for nature to produce large copy numbers. And mathematically, it is interesting to understand what define these numbers. And so what large number are doing for us? Well, without this large amount of number, maybe the action will not be possible. So let me state the second thing. When you have a large number, a large copy number, it's not necessarily a waste. We can imagine that you have a lot of molecules, a lot of cells, so you can take some of them. But actually, these large numbers are necessary and should not be seen as a waste of nature, because in fact, in the majority of cases, the function which must be carried out is to find a small target or a small hole, the position of which is not known in advance. The change of nature is therefore, the chance of nature is therefore compensated by large copy numbers. In other words, if you have to find here, and I will explain in the next slide, if you have to find a target and you have a source, and the source emit particles, which I'm going to draw now in a different color, in red. So if you have large copy numbers and they don't know where is the target, then you need a large amount of them in order for one of them to arrive on time. And if you remove a certain amount of them, you remove this because you take them out of the process, then it may happen that you don't find the target on time and that the process you wanted to activate is not going to be activated. So I would like then to illustrate this idea again. So you have, let me go back to the color I've chosen. So this is, you have a source. Here you have a target. So for example, suppose the source is where molecules or ions are produced. So you have many ions here. And in between, so this can be, for example, on the surface of a cell, inside the cell, you can have obstacle. And so what can happen is that you want to have these molecules that are generated here. You want this molecule, when they are generated, to move. And this depends, of course, on the type of motion, but you want one of them at least to find the target. And when the target is found, a process is activated. So you activate something. So for example, you generate here calcium ions, you generate any kind of important messenger molecule, such as IP3. It can be also a G protein. So that you name it. The biology is full of this example, and you need to go to this target, which can be, for example, a channel. And when the channel is activated, that's it. You have a flow entering inside the cell. So here you have a flow. You have a current flow. And so mathematically, the question becomes how long it takes for this molecule to find the target, and the time it takes depends on the distance between the two. So here it would be the geodesic distance. You have to go, you have to avoid the obstacle. Let's call it delta. For example, if it's a diffusion process, it depends on the diffusion process. Log of the number of particles. So which is interesting. What is interesting here is that even if you have a lot of them, you still it will take some time because of the log, and because the molecules are really generated far away from the source. If there is a possibility to generate something much closer to the source, which depend on a sort of a gradient, then the time becomes delta square. But instead of having log of n, you can have n to some power, let's say power alpha, where alpha depends on the type of the gradient that you have, which can accelerate the process. But this is not biological most of the time, because things are generated at the source point. So let me continue. So to recap this is important in signaling transduction. This is important for synaptic activation. This redundancy principle occurs in many different type of different cells, such as neuron in the brain. And I would like to discuss here these fertilizations, and also what are the laws. What can this telling us about how nature compensate for hidden targets? So the effect of the geometry. So again, as I said, as I showed previously, you want to make sure that an event will occur. So this event has to occur, and so the question is how long it will take. As I showed to you, it depends on the log of n, and to compensate, you need large number, and that's why in many cases, like in fertility, you need many copies of the same object generated at the same time. And why is that? Because they are constrained in time. You want to make sure that the event occurs in less than a different time. So if you have a source somewhere and you have a target here, you want to make sure that the target is going to be activated, let's say, in less than a few milliseconds. And this is, for example, very important in the case of our transduction, sensory transduction, when you want to, if you have an animal and there is a prey or predator that is going to kill the animal because he wants to eat it, in order to defend itself, you first have to see the animal coming to you. So then, in the brain, the mechanism should be very fast. And one way to accelerate is to create, when you have, for example, a photon that is generated in the eye, you have to generate a second messenger that will open here, for example, a channel very quickly. And this depends on these numbers, the number of copy numbers. And sometimes they're not generated immediately. They could be generated with a certain time scale, which is also going to affect, but not that much, the mean time. So two things are important, is the geometry, where this is occurring, and the nature of motion. Nature of motion. If it is Brownian plus drift and so on. So this we have discussed in the previous slide. This is an example of a dendritic spine where you have calcium. The calcium has to find here a specific target in order to open here a mechanism called calcium-induced calcium release, CRCR, where you have an avalanche of calcium. And the first one to arrive can lead to an acceleration of the process, basically a factor 10. It's 10 times faster. Again, this is the same thing here. You have many IP3 molecules that have to come and open the IP3 receptors. And again, you have here many this is a synapses, so neurotransmitter are going to be released here at the vesicles, and this is the blue trajectory that you can see. Some of them leave, but if they want to activate here a receptor because of the specific geometry, what is very interesting is that the number of the order of 3,000 of neurotransmitters what is very interesting is that they compensate the low probability, which is of the order of 10 to the minus 3, to bind here the umpire receptor before exit. So the two together combined makes a real probability of something which is close to 1, making this event surely. So this for surely, when you'll have a vesicle that is released, it will open some channel. So how nature has selected this number, this is something that we don't know. And I would say another example is the case of selection of an antibody during the immune response, where you need to have 3 elements, which is called V, D, and J. So V, D, J are first selected, and then V and DJ has to they have to find each other. So this is here illustrated in this process where you have chromatin, where you have many V in green, and you have one DJ in red, and one of them by Brownian motion. This is a polymer mechanism, polymer Brownian dynamics, where you can see in here the two. After some time they bind, and this is a way to select which type of antibody receptor will be expressed on the surface of the cell. So this is shown here. You will select by this one type of receptors, and so the time it takes is very fast because you have already many of them, and it's the first one to do this not only increase the time, but also it's a process of selection. And so the most evident and well-known case is the case of fertilization, where you have many spermatozoa entering here into the cervix, and you see a trajectory in green, and they have to find usually the place of an ovule located somewhere between the beginning and inside the fallopian tubes. And this is an example here of simulation we made a couple of years ago, where if you have many trajectory and if you look at the first one to arrive, this is the distribution of the one arriving, then you see that you select these very defined trajectories. They are shown here in dashed lines. So this is the what we call the extreme trajectory, the extremal trajectories, that where you have really the sperm shooting towards the destination. Although many of them are going to lose themselves, they are going to move around, but not necessarily finding the target. Let me draw here an example of a trajectory. This is a non-optimal trajectory that will move, but this is very far from the shortest one that are going to be selected here. And what was interesting in that case, I would say, is the fact that if you plot here the volume of the uterus, trying to understand what defines those numbers, if we see how if there's a dependency between the volume of the uterus and the number of spermatozoa, as you can see here, there is a trend. There is a trend to say that because it is very hard to find a small target, if you have a large volume, then you need to compensate this by a large number of spermatozoa. But the evolution explanation is still missing to understand this phenomena. So to conclude, I would like to say that using large number is not a waste in nature, as has been seen so far, and this is why I wanted to insist on this theory. It allows to transform rare events into sure ones in a reasonable amount of time so that the function can be performed. The number of copy is defined by the geometrical organization and the nature of the motion and a small target or small targets if there are many. So you need three things to define that three things are defining these copy numbers. The organization, which is the geometrical organization, the type of motion, that can be Brownian motion, or any kind of random motion plus deterministic motion, and the fact that you have small target and the molecule they don't know, or the cells, they don't know where the target is. And I would conclude by this, which I think is very important. It's our relation to nature. We should integrate this redundancy principle, especially in defining what we define when we say quotas, how many things we should leave and how many things we should take from nature. We have no idea of any unraveled goals where large numbers are used to compensate this hidden geometry and a function to be made with a specific time constant. And thus we may sterilize a lot of necessary processes if we don't integrate in our understanding the fact that large numbers are not a waste and we cannot just take. We have to understand how much we need to take and how much such a process needs in terms of this large number. So these are some references. The first one is where we publish in a very large review this redundancy principle in Physics of Life review, plus some, so this was here comments that we wrote. This is the review itself. This is another example where we have we communicate on this. We have also mathematical references which can be found on internet. The case of spermatozoa in this Journal of Mathematical Biology, and I recently in French wrote a general text about what defined short time and long time in biology, summarizing that it has to do with the shape, the motion, and small hole. And I thank you for your attention.